All right, so while we are waiting for others to join us as the meeting progresses, we are going to quickly summarize from where we stopped last time. So during our last session, we were able to, yes, we got these results. That was the result we got from the result we got from our docking. I hope my screen is visible.
Yeah, it is visible now.
Okay, sir. So we got these results from the docking, and from that docking we saw that 129.125.621 had the highest binding affinity of minus 9.6 or better, has the lowest binding affinity of minus 9.6. So compared to our controls and the standard, from there we're able to create a table that looks like this, where I said that this is how the results table will be. We must have seen other docking literatures. So all what we just need to do is to copy this CID code and get the name. Then we'll fill up this ligand column with the names of the compounds with their respective CIDs. So we went further to also generate their superimposition image. So we saw how our ligands we are well superimposed with the native ligand, which depicts that the interaction actually took place at the same binding coordinates with the reference native ligand that defined binding site of that protein. So it was a well-performed interaction. So that compound that gave that high binding result, we'll give a kudos to that compound. And then we went further to generate the 2D interaction. So this is the 2D interaction of that compound. So we are going to just look at this now. From this 2D interaction, you are seeing that the major interactions here are van der Waals, conventional hydrogen bond, the alkyl and the pi-alkyl. In fact, it will be difficult to differentiate alkyls and pi-alkyls. So both of them happens to just be the same. So there are two major classes of interactions here. We have the hydrophilic interactions and the lipophilic interactions. So the hydrophilic interaction here is the conventional hydrogen bond, which is the water-soluble interactions, and it's very good that we have such kind of interaction, because if you are designing a drug and the drug does not have that conventional hydrogen bond interaction, it means that the interaction at the molecular level will be very poor. So irrespective of the kind of interaction that you must have, a good drug must also have a conventional hydrogen bond interaction. Then right, the other bonds are the lipophilic interaction, which is also good. You must balance the hydrophilic and the lipophilic interactions of your drug. So we are seeing the ones on green, which is the ASP bonding with the hydrogen bond from the amine terminal, from the terminal amine group of this ligand. Then the ones on light green are the van der Waals forces according to the key here. So this one van der Waals forces, their job is just to stabilize that binding environment, the binding interaction. But the hydrogen bond interaction helps for specificity. It tells us that the interaction was well specific, which means it actually targeted the receptor, while the van der Waals forces just tells us about the stability. So they help to stabilize the ligand at the active site. Then the acyl and pi-acyl, they are all lipophilic interactions. Even the van der Waals too is a lipophilic interactions. So that's acyl and pi-acyl here. The job is also to balance the interactions with the hydrophobic chains of the proteins in the amino acid and that of ligand. So there must also be this lipophilic interaction must also be there. Yeah, there must also be there because not all the amino acids in the protein are hydrophilic. So some of them are lipophilic. So we also need to see that balance interactions. So that is the whole idea of these 2D interactions. The 3D just showing us what we have already in 2D. So the 3D just showing what we have already in the 2D, but just keeping it in a 3D dimension. And so this is showing, remember, this is hydrogen bond cloud. This is hydrogen bond surface. So we can see how the ligand was, how the ligand, the position of the ligand at the binding pockets. So this hole here, where the ligand is inside, you can see ligand inside, is also showing the position of the ligand at the pocket site, at the pocket site of that protein. So there is really not much in here. When we look at literatures, we can see different ways of interpreting our results. So tonight we'll now look at the ADME properties of this our heat ligand. We might even look at the first two. I will just pick the first two, then I will assess the ADME properties of the first two. All right, are we together?
Yes, sir.
Yes, sir.
All right, so I will look at the pharmacokinetics properties of our heat ligand, the first two ligands. Then we'll see if this top ligand, definitely you know that we have done some preliminary screening. We have done draw likeness screening, and draw likeness screening also contribute to the pharmacokinetics properties. So that may not be an issue for us. It simply means that any compound that arrives at this stage is drug-like, because we used the drug-like properties. So I was really afraid to see if we may, after we have filtered a lot of compounds out, if we may end up still having the ones that will give a better binding result. But at the same time, I was not also afraid, because we did not just rejected those compounds by will. There was criteria, and the criteria was drug-like properties. So that simply means that those compounds that are past that stage of drug likeness filtering, must give a good binding affinity on or with the protein or receptor to be used. So it's a way of screening off some of the compounds instead of you wasting your time docking all those compounds at a time. So we shall take this and go to PubChem CID, go to PubChem to get the CID, or to get the SMILE code, or I can just come to one of, I think we have that Swiss, that we have that table that has their SMILE code. Okay, the natural product like that. So I think we have this table. So I'll quickly trace their SMILE code from this table. Oh my God. So this is the 12129. Okay. Let me be sure of the number. So 12129125621. So I will quickly trace it. So that is number 16. So I will just copy this SMILE code now. So we can look at the ADME properties. We'll go straight to ADME lab. So we can quickly go straight to ADME lab. At the ADME lab, you're supposed to respond as quick as possible. Okay, it's coming up. So we'll quickly look at the ADME properties. But we'll not focus on the toxicity again because we have already looked at the toxicity previously. And it is, from our criteria, is assumed that this compound is not toxic. Right, so I shall look at the ADME details. So this is the we are focused, our major interest will now be on the absorption parameters. Today, that is what we want to deal with. Please, if your mic is on, just kindly mute your mic. So we look at what is the implication of these absorption parameters, CACO2, NDCK, PAPA, PGP, HIA. What are their implications? We look at the distribution, their implications. We look at the metabolism, their implication, and the excretion. Please, if your mic is on, kindly mute your mic. So these are what we shall study tonight. So you can quickly download the result as well. Click at CSV and download. So you can prepare the table. You can now prepare your table for your pharmacokinetics table. Here you pick the ones that are absorption, you pick the ones that are distribution, the ones that are excretion and metabolism. Literatures will still help you to guide you on how to present the work. Let me prefer transposing this. So let me just transpose this so it will be easier for us to view. All right, so it is better now. So at this point, you can easily see your properties. You can now pick out the ones that are absorption, the ones that are distribution, the ones that are metabolism. Use them to create your table. So this ADMET, the ADMET website can help you to select absorption. So these are absorption, CACO2, MDCK. From this to this are under absorption. So you can keep them as absorption while creating your final table of your results. Then under distribution too, you pick PPB to BCEP inhibitor. So that is just what you will do to arrange your table of results very well. That's what you do to arrange your table of results very well. So, but our interest is the explanation of those parameters, like this FU to this. I think this one's on red. They are distribution parameters. Why the cytochrome P enzymes are your metabolism parameters. You can keep them with different color. Then these clearance plasma and the half-life, they are excretion parameters. And keep them on green. So you see that you can now use it and create your table, while your CACO2 to this, they are your absorption parameters. So we have absorption on blue, we have distribution on red, we have metabolism on dark blue, and the green are the excretion parameters. So what are the implications of these values? What are the implications of these values? And remember, there are lots here, but we pick the ones on absorption and distribution. So tonight we'll now look at how can I interpret these values. So this CACO2 of minus 4.99, what does that imply? The MDCK value of minus 4.7, what is it telling us? The Pampa value of 0.0. So what are all these values telling? What can we deduce from these values? That is what the BBB value, what does it imply? So that is what we have started to do tonight. So these are your pharmacokinetics properties. You take your time to study them. The whole of this explains absorption. When you summarize this whole result, we tell you how well the drug can be absorbed. The ones on red summarizes the distribution. These are the distribution properties. So when you do a critical study on them, you're able to know how the drug can be distributed. Then the whole of this now on dark blue now talks about the metabolism of that drug. So when you do a critical study on all of these, it gives you information on the metabolism of the drug. While the one on green now, these two talks about the excretion of the drug. Does the drug stay long in the body, or it can easily go off? What's the half-life? So the half-life of 0.54, what does it imply? That is what we want to do tonight. And at the end, that will be the end of pharmacognosy training. While we have already started, that of green chemistry, which tomorrow will be our second session. So I'll quickly share my screen for what I have to do with us tonight. So tonight we are looking at understanding pharmacokinetics properties. So can you see my slide?
Yes, sir.
All right. So understanding pharmacokinetics properties. when we talk about pharmacology, pharmacology is in two phase. In fact, drug is in two phase. How the drug reacts with the body, that is the molecular docking, the pharmacodynamics. How the drug will interact with the receptor, is we're talking about the efficacy of that drug. But one thing again that makes drug, in fact, one reason why most drug fails is also how the body interacts with the drug. That is the kinetics, the pharmacokinetics. It now explains how the body will interact with this drug. Will the body welcome the drug? So these two must be put into consideration. They must put into consideration in drug design. So we shall start with the absorption parameters. We'll look at all of them, those ones in the ADME result. We'll look at all of them and their implications so fast. I have a lot of slides here. I will just run them so fast to make sure that we conclude tonight. Right. So we have about 1 hour, 30 minutes to run down these slides. So the first one is CARCO2. CARCO2, what it is CARCO2 is an in vitro model of human intestinal permeability. So it tells you how the drug can be absorbed by the human intestine, how it is able to permeate the human intestine. Of course, stand up for this in silico tools. It's just like what we discussed yesterday for those who were in the webinar, predictive modeling. All these are all predictive modeling built by experimental data. Initially, they were trained. The model was trained with experimental data, so it can now predict. Can now use it to predict based on similar information you have given to the model. It can now tell you, see what it could be. And the reason is that predictive modeling now helps us to even perform those kind of experiments that are difficult. Tell me how you be doing CARCO2 in the lab using animal models or what. So thank God for all this. And I'm even wondering how drug was discovered without these tools. And now that we have these tools, how many drugs has also been discovered? So what the CARCO2 does is an in vitro model of human intestinal permeability. Now the implication is that a high value of CARCO2 means good intestinal absorption. So if your drug has a high value of CARCO2, then it is a good result. So when you have 5 compounds and you subjected these 5 compounds into ADME properties, so the one with higher CARCO2, maybe you have 5 hits, and you took these 5 hits to your pharmacokinetics studies. So you now see that the one with higher CARCO2 implies a better absorption, while the one with low CARCO2 has poor oral absorption. So the next one is MDCK. So just as we have seen in the ADME result, MDCK is the same model for general membrane permeability. It is synonymous to the BBB prediction. So it also talks about, see, everything is about permeability, absorption, to penetrate. So the implication is that a high value also implies good membrane diffusion, possible tissues, and possible CNS penetration, because this is similar to the blood-brain barrier. And although we are not likely to be exactly blood-brain barrier, but it has the ability to permeate some membranes, and some of these membranes are the CNS and sometimes the PNS membrane. So a low value means poor permeability. So look at this when you have your results. Look at this, it will guide you. Number 3 is the pamper. So the pamper is an artificial membrane assay that measures the passive diffusion. Still talking about diffusion, all of them are pointing towards absorption. So high value means strong passive absorption. Low value means weak passive mobility. So when you compare them, you will now know the PGP inhibitor, PGP is also another absorption parameter. It predicts whether a drug can inhibit the pre-glycoprotein with efflux pump. So we have the PGP inhibitor and the PGP substrate. So here, if a drug has a high PGP value, it may increase drug interactions and toxicity risk. So that means that drug has ability of inhibiting the pre-glycoprotein. So look out for this. It means that in our absorption results, a good drug should have a low, a negative PGP value. So a negative value indicate lower interaction risk. Then the fourth one is the PGP substrates. It predicts whether a drug is pumped out by the pre-glycoprotein. The first one is if a drug inhibits the protein, but now if the drug can be pushed out as a substrate, can be pushed out by that protein. So a positive value means reduced absorption and lower bioavailability. So when you have a positive values, it means there is a reduced absorption. So it also implies lower absorbability. But when you have a negative value, that means better intracellular retention. So it simply means that the drug is not pumped out, so that the drug is retained. The drug is retained within the cell. So the drug is retained. So when you have a low value of PGP, that is a good result. Then the other parameter is the HIA, which is the human intestinal absorption. So these are parameters that talk about absorption. What it is, it predicts the percentage of drug absorbed in humans, the percentage of drug that is absorbed. In fact, only the HIA is even very, very important to talk about absorption parameters. The implication is that high value means good oral bioavailability. So if a drug can have a high value of HIA, that implies there is a good. Remember, all these things are based on the structure. All these parameters are generated based on the structure. Just looking at the structure, because whatever a compound can do is as a result of its chemical structure. Yeah, whatever a compound can do, whatever a molecule can do, is as a result of its chemical structure. So when you have high HIA, that implies a good value. It can easily be absorbed. Then the reverse is the case. Then we have F20, we have F30, F50, and so on. The F is also predicting oral bioavailability. Yeah, is predicting oral bioavailability, and this one is telling you the probability of greater than or equal to 20%. So the ability of absorbing about 20% of that drug. So when the value is high, that means there is acceptable minimum absorption. So there is an acceptable minimum absorption at 20%. There is, I think, is absorbed. A lower concentration of that drug can be absorbed. Then low value means very poor absorption. We have F30 is still looking at 30% of that drug, at least 30%, and it still takes about high means moderate oral absorption. So by the time you look at F20, is very is very good. You base your work with the F20, which tells you the absorption at a minimum of 20%. So, and let's take a drug to be 100 grams. So we're looking at can 20 gram of my drug, at least 20 gram, be absorbed. That is what the F20 is looking at. Can 20 gram, at least 20 gram and more of my drug be absorbed? So if you have a higher value of that F20, it tells you that a good amount of this compound can be absorbed. Is orally bioavailable. But F30 is now looking at 30%. So if it's high, that means moderate oral absorption. If it's low, suboptimal bioavailability. Then we also have the F50, which is the probability at 50%. So we are saying, let me look at 50% of the drug I'm taking. So if we're looking at the 100 milligram of the API, so we are saying, okay, can you absorb at least 50 milligram of the API out of the 100 milligram? So this can tell. So when you have this value, it means excellent absorption, because now you can see that a good amount, a high amount of that drug can be absorbed compared to when you're focusing on 20%, looking at 20% of the drug. But at 50%, now it means that how we should even have testing, you can have F80 to say, oh, can at least 80% of the drug be absorbed? That will even tell us that the totality of that drug can be absorbed. So a higher value of it also implies excellent, very good oral bioavailability, lower value, poor oral performance. So that is that for distribution. So I just want us to, I'm just running down everything about that. Then coming to distribution now, we have the first one, OATP TP1B1, that is organic anion transporting polypeptide 1B1. Is a liver uptake transporter responsible for hepatic drug clearance. So it's at the liver. It tells you if drug can be cleared by the liver. The implication is that if the value is between 0 to 0.3, it has low likelihood of that interaction. It means that the transporter, it finds it difficult to be cleared by the liver, so it can be retained in that liver. But 0.7 to 1 means high probability of substrate inhibitor, so possible hepatic accumulation of drug interaction. So better to say, put it this way, if the OATP1 value is low, it means that our drug is not interacting with our liver. And we know that we did one of these parameters for toxicity, which is a hepatotoxicity, that claims if a drug can affect our liver. So when the drug is having better interaction with the liver, so the drug have a tendency of affecting that liver. So a low value means that there's likelihood of interaction with the liver, and then a high value means a probability of a possible hepatic accumulation or drug interaction. So it's better you have that low value. That means there is no, that likelihood of interacting with the liver is not there. So at this point, we're expecting a low value in OATP1B1. The next one is the OATP1B3. Is still hepatic transporter similar to the one we just discussed earlier, it still mediates drug uptake into the hepatocytes. So still low value of this still suggests minimal liver transporter interaction, and high value still suggests DDI risk, that is drug-drug interaction risk. So we should always be careful. We are expecting to have low value in OATP1B3. We have the BCRP, the breast cancer resistance protein. This is the efflux transporter limiting drug absorption and brain entry. So it tells that, okay, I'm limiting the drug from entering into the brain. So I don't want the drug to be distributed to the brain. So you look at it. If it has a low value, that means low value means better tissue penetration, but high value means reduce the bioavailability. So the job of this man is that it stops those compounds. It limits how the drug will be absorbed into the brain. So it limits drug absorption and brain entry. So if it has a low value, very low, that simply means that there's a weak efflux, there's a weak penetration. So, but if it has a strong value, high value, I mean there's a strong penetration. There's a strong penetration. And once there's a strong penetration, there's reduced bioavailability. So we are saying that the BCRP should have a low value. And once the value is low, it means that that drug absorption into the brain is limited. So is limited. But when the value is high, it means that that drug can penetrate, can be allowed to be distributed to the brain. So expect a low value for BCRP, better distribution. We have the BSEP, the bile salt export pump, liver transporter involved in bile acid secretion. Low value means low inhibition risk. So we're also expecting low BSEP value. And the high value here also means possible BSEP inhibition. That implies cholestatic liver risk. So in summary, we are saying low value for BSEP. Still on distribution, we have the BBB, the blood-brain barrier penetration. Ability of a drug to cross into the CNS. Of course, if it's low, below 0.3, there is a poor BBB penetration. That is very good. 0.4, 0.6, moderate CNS access. Now, this is, we only encourage having high value if we are creating a drug that can manage neuro disorders. But if the drug was not meant to manage neuro disorders, it means that if it penetrates into the brain, into the neurons, into the central nervous system, since it was not meant to interact with them, if it finds its way there, it will lead to adverse side effects. We don't want these compounds to permeate our brain. So higher brain penetration is, like I said, it's only useful for CNS drug, but it's risky for non-CNS drug due to CNS side effects. So a low BBB is good. A lower BBB value is good, except you are creating a drug that should interact with the CNS or with the brain. Another parameter is the MLP1, the multi-drug resistant protein 1. Yes, what that is it's about the efflux transporter causing drug excretion from cells. Yeah, if is low, there's a minimal efflux, a lower movement. But if it's high, high efflux, so high movement. Once that movement is high, it depicts that reduce intracellular drug concentration. So these drugs should be there. They're supposed to stay at the cellular level. It's supposed to be there. But if there is high excretion, a high movement away from the cell, that means that the resistance risk should be high. So the drugs are supposed to stay long at the cells before they leave. So a lower value is also okay for us. Then the PPB, the plasma protein binding, is a fraction of drug bound to the plasma proteins. Lower value means that some of these drugs cannot bind to the protein, so more free drugs are available. So these are just, let me not call it absorption parameters. They are not by absorption, they are predictive. So rely on them up to 80%. The only thing is that we may not tell the model how accurate the model that was used to build them. And that is why if you do ADME with, ADME with ADME lab and do ADME with other, maybe ADME SAR or Swiss ADME, there will be variations in their value. And some of the variations self might even make you to begin to question which one should I use, because you cannot tell the validity of that model. And that is why we are training researchers to build your model, learn to build your model. So we thank God for what Dr. Olabiwotimi did to us yesterday. Our bones, we are all broken yesterday, and there was a new resolution which we have accepted the call, and we are looking we're looking up to starting up something. So a low value means they are free drugs, but high value means that these drugs can actually bond to our plasma, and that will not be good for us. Low free drug fraction and longer half-life, so that with possible displacement interactions. The log VDS, which is volume of distribution at steady rates, it's about the extent of drug distribution into the tissue. So about the flow rate, the speed at which the volume of the drugs, this amount of the drug is entering into the tissues. We are saying that low value means that small amount of these drugs enters the tissue, so that will not be good. That means some of them are just found at the plasma. They got bonded to the plasma. High value means extensive tissue distribution, right? Another one is the FU. The FU, that is fraction on bond, the fraction that did not bond to the plasma protein. So fraction of drug not bound to plasma protein, that is what I just said now. And that usually gives us the pharmacologically active portions, because we don't want this protein, this compound, to bond to the plasma protein. We want to have free drugs in the cell. So when the value is low, it means there is a high binding with the plasma protein. But if the value is high, so there is greater activity. Yeah, because when we have high value, that means if you have high FA value, that means a good number of the good fraction of that drug did not bond to the plasma protein. So we have large unbound portion. So that means a good amount of the drug is free, and that ensures greater activity. But at the same time, since they are free, they will have high mobility. They will have faster clearance. So that is just the implication. But we are looking at high value of FU at this point. The FU should be very high. We can look at the metabolism. think I'm a bit fast, but that is just how I really want it to be. So we'll run down all the parameters, then we talk, we interact so far on what we have done. If there are challenges so far that you have encountered during the course of the work, and we summarize the training, so we draw the curtain close on macrognosy. Metabolism parameters, of course, all the metabolism parameters, you're just looking at the cytochrome P450 enzymes that are responsible for drug metabolism. So we are checking, can they be inhibited? This cytochrome P450 enzymes, can they be inhibited? Can the drug inhibit these enzymes? That is just what our focus is. So we have the CYP, the PA, the 1A2 inhibition, which is the probability that the compound inhibits the 1A2 enzyme. Could it be good? So a lower value means low inhibition risk. High value means possible drug-drug interaction, which is not good. So this enzyme should not be inhibited. Yeah, should not be inhibited. So we're expecting low value, very low value for CYP1A2 inhibition. Then CYP1A2 substrate, probability that compound is metabolized as a substrate that can get metabolized by the enzyme. We are saying a low value means minimal metabolism. A high value means likely metabolized. So that means the inhibition and the substrate they should be in opposite direction. We don't expect the drug to inhibit the cytochrome P450 enzymes, which means that there should be a low inhibition value. But when it comes to substrate, which is the ability of the enzyme to metabolize the drug, ability, because that drug is now a substrate, so the enzyme wants to metabolize the drug. So a high value of CYP1A2 suggests that that drug can be metabolized by that enzyme. So this ideology can cover to all the cytochrome P because they have inhibition, they have substrates. The same thing to this place. We don't expect to inhibit this enzyme. So a low value means safe. That means it's okay. But for the one of the substrates, we are expecting a high value. We are expecting a high value. High value means that there is that it can be metabolized. So the one of these enzymes are for drug metabolism and DDI for drug-drug interactions. So we are all saying that all the cytochrome P450 inhibitions parameters, all the cytochrome P450 inhibition parameters should have lower value, while all the cytochrome P450 activators, like the ones of the substrates, should have a higher value, while inhibitors should have a lower value. Here too, cytochrome P2C9, we are expecting that a low value is good, minimal interaction, as minimal inhibition is good. But a high value is a very rich of toxicity. So very rich of toxicity. Then the substrates, like we have just summarized all the metabolism, we are expecting that a high value is very good. A high value is very good. Here too, cytochrome P2D6, we are saying low value is good. That is low DDI consign, low drug-drug interaction. But for the substrate, a high value is good for substrate. A high value is very good. High value is good. So that is all we are saying. 3A, 4A inhibition. a minimal DDI consign also. So we can run this down 3A4 substrate. We're expecting that a high value is good. High value is good. So that is all what you can get from metabolism. I think this will just help us to understand all what we need to know. So cytochrome P450 enzymes are very, very important. They are very, very, very important. We are saying allow them to break down your drug. Allow them to break down your drug, because if this drug refuses to be metabolized, they will interact with other drugs. That means they will remain there. They're going to remain there. And when you take other drugs, they have the ability of interacting with other drugs. Mostly, if you are taking, I don't know why our laboratory sector gives us MPY that everybody must have malaria and typhoid at the same time. So if you are taking these drugs simultaneously, and you saw that the cytochrome the cytochrome P450, we are not able to metabolize these drugs, this ciprofloxacin, and your artemether. So if they are not being well metabolized by this enzyme, they will remain there, and as they remain there, they will begin to interact with themselves, which will lead to other side effects. So that is the whole idea of the metabolism parameters. So we shall see how many they are. They are very, very much. They are very much about 14. Yes, I think they are about 14. This is the last one. The human liver microsoma stability. It predicts metabolic stability in human liver microsomes. So a low value of this implies rapid metabolism. Of course, once the metabolism is high, short half-life. All right, so we shall look at excretion parameters, which is the last parameter. The excretion parameters, which is the last parameters. We have the CL, which is the plasma clearance, and that is saying that it predicts the rate at which a drug is removed from the bloodstream, excretion, via liver metabolism and or renal excretion. So if you have a low value of the plasma clearance, it means that drug is not easily removed. So the drug remains longer in circulation. And once the drug remains there for a long time, there will be risk of accumulation and toxicity. Yes, so that is telling about the clearance, the excretion of that drug. But if the drug has a high clearance value, it means rapid elimination, that drug can easily be removed from the body. So, and because of that, now you notice that you don't take only one dose in the morning. You have to use this parameter to define dosage, since it has rapid clearance ability, so it doesn't stay long in the body. You can take twice in the day, and that will be better for you. So the implication also implies that high clearance reduces bioavailability, half-life, while low clearance increases systematic exposure. So we are, even if we are encouraging low clearance, but let's also bear in mind that will lead to some risk effects. Right, the last one, which is the T0.5, that is half-life. The half-life is time required for half of the concentration of the drug to be reduced. So if I'm taking 500 milligram, So we can just estimate the time it will take for half of that drug, 250, to be absorbed. That is half-life. So a shorter half-life means rapid elimination. So if the half-life is 2, another half-life is 10 minutes, it means that it will take 2 minutes to 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 absorb, or it takes 2 minutes for half of that compound to be eliminated. But if my half-life is 10 minutes, it means that it will take me 10 minutes for up to half of the API to be eliminated. So more of that compound should be there. So we are expecting high half-life, that is prolonged action, risk of drug accumulation. So at the same time that your drug, we are saying, okay, let it remain, let it stay for a long time. There is high risk of accumulation, drug accumulation in other parts of the body. So we have come to the end of pharmacokinetics discussions. I think this is exactly an hour now. So we shall take questions, if any, and look at for what is expected from us and do a summary. Thank you very much. So let's take questions if there are. Yeah, let's see your questions. Okay, Victor, you can go ahead.
Yeah, good evening, sir. Thank you for the wonderful lectures. So my question is, okay, this one is the one that has the highest binding affinity, and talk about the drug likeness. So what I want to know is that what if all those parameters, this absorption, excretion, and metabolism, does not meet up with the standard? So does it mean that we can discard that molecule? Because I'm just seeing some disparities in that readings. Thank you.
Okay, so if you have actually checked, you see, you checked those parameters, and you have weighed it, you did your weights, and you see that this my compound. And also, this is the reason why I don't work with the first compound. I use first 5. So you can compare the first 5. In as much as the first 5 is giving you better results, so you take the first 5. That's a lower binding affinity. So once you take this first 5, you now compare the ADME results. So you will now see that, oh, it may be possible that, like you said, the one you are looking at may fail these parameters. What do I do? That is where lead optimization comes in. Yeah, because the essence of lead optimization is to have better compounds that will have improved ADMET property. That is why we are bringing in a lead optimization. And like I told us in one of our training, for those who were privileged to join us, that in lead optimization, you are modifying the structures by bringing in some macrophoric features. But the best approach to lead optimization is, when you just modify the structure to have a better ADMET, it will tamper with it will definitely tamper with the binding affinity, because you have changed the structure. But the best way now is to use your bioisos test. That is the training coming forward by March. Yeah, that is the training we are putting on by March. So I can now modify my my compound because it failed in the ADME properties. I can now modify my compound such that it will still maintain that efficacy. It will maintain that efficacy, but the difference is that it will have an improved ADME property, but it will maintain that same efficacy, and it will have an improved ADME property. So what we do is to locate some of these functional groups that maybe that we are responsible for that poor ADME result, or for the toxicity. Then we just replace that functional group with another functional group that will still maintain the efficacy, the binding affinity. So even when we go back for docking, we will still observe that similar binding result, but now with improved ADME property. So that is what you will do. And this thing I just explained now will be included in our training coming forward by March on lead optimization. So we are doing that. So these are what we have for that March training. The work is focusing on integrating pharmacophore with molecular modeling and molecular docking. So we shall do pharmacophore modeling. We shall do feature-based screening, toxicological screening. Of course, we must always have a toxicity screening. Then we are doing lead optimization, where we bring in bioisostates tests. Very important. Then pharmacophore recombination. Yeah, we did not do that, but this pharmacophore recombination is also another approach we can apply in the optimization. Yeah, it's another approach we apply in optimization, where we can divide a compound into some part, collect part of this compound, create part of this compound, and build our own structure. That is pharmacophore recombination. Is widely acceptable in drug design. These are new techniques we use just to make sure that we achieve compounds that will have better ADME properties. Thank you very much for that wonderful question.
Before we go further, I just want to say something about the question just asked. I don't know if I can have the floor.
Yeah.
Yeah, the features that we've looked at, the ADME features that we looked at, from absorption to elimination. When you are talking about them, we are categorical about some of them being bad and some of them being good, depending on the value. And probably that is why that question came up. Those features, those drug features, especially the ADME features, their value can also depend on what you will want a drug to achieve. For example, a drug that has a poor permeability, yes, predicts the fact that, the oral absorption is probably not going to be good. But that does not preclude that drug or that candidate from being a good drug. For example, we can also use drugs, through parenteral meal, intravenous, for example. And there are many polar drugs that have very poor membrane permeability that are also good, that are also very valuable drugs, especially antibiotics. And even this poor permeability even comes as a very good attribute to drugs that we want to have to spend a long time in the plasma, in the blood, for example, in the treatment of septicemia. So for those drugs, we want them to stay in the blood for as long as possible so that they can help us to take care of our infection. And the good thing about those polar drugs is the fact that once they are in the blood, they don't permeate membrane, which means that they don't distribute into other organs that we don't want them to distribute into. Organs like, the brain that you talked about. So again, that a drug, for example, and again, apart from oral absorption, even in terms of metabolism, there are some drugs, for example, that are prodrugs that we want the liver to take up and combine and convert them to our active drugs, because the biotransformation takes place in the liver. So for that kind of a drug as well, you want, the liver uptake to be very good. Yes, there's a risk of, interaction with the enzymes, and that can also predispose to drug-drug interaction. So again, what I'm trying to say is that, yes, you lose some, you gain some. So it is now depends on your ability to be able to manage these attributes for you to have, your drug that will have a very good value. And as I said, it's all related to what do you want to achieve with your drug? What kind of activity, is expected of your drug? So you can't use a one rule fixes it all. It has to depend on what you want to achieve with your drug. For your drug that you want to, by all means, administer orally, of course, you have to have a good oral bioavailability. For a drug that is a prodrug, you want it to have good absorption. For a drug that you want to use to treat brain diseases, you want it to have a good blood-brain barrier permeability. For a drug that you are afraid of its neurotoxicity, you don't want the blood-brain permeability to be high. So again, as I said, these are need-based, and we cannot use just one rule fixes it all here. Thank you.
Yeah. Thank you so much, Dr. to me for that insightful contribution. So it all now depends on the roots. What kind of, how you want the drug to be administered, how you want the drug to operate. So you look at for the parameters that we adequately fit into what you want. All right, thank you very much. Okay. We have other question.
Hello.
Yeah.
Yeah. Well done for the training. So I have a question which is outside the scope of tonight training. So it's an question that borders about the in vitro. So we did molecular docking by binding ligand with protein. So I want to ask, because I'm not a chemist, so I'm a microbiologist. So I want to ask, assuming I want to look for a receptor that will bind to my drug. So which method will I use? So if it is an in vitro experiment.
Okay. If you want to now do in vitro experiments, then you have to look for their assay. Look for their assay. Yeah, look out for their assay. They are called assays. Now, like for instance, if you want to do cancer work, you buy these cancer cell lines. You have to look for the cell lines. They have been a lot of work has been done on them. The only issue is that you may not be able to have access to all because they are expensive. Some of these assays are actually expensive. The biomarkers are expensive, but they are, because you don't need, you will not use that particular receptor now. The operation does not work with that same particular receptor you use in your docking. But you can look for assays that has similar genetic information with the protein you have used to dock for proper confirmation. And that is also why you don't use one single protein. You can use multiple proteins that interact with that same target. That one one pathway you're working on. So you look for multiple proteins that has associations with that pathway. So your in vitro studies, you buy assays, buy biomarkers. They are available in the market, but they are expensive. Seriously, to conduct in vitro experiments in some of these diseases, some of these diseases are actually expensive. That's why if you look at the most common in vitro experiment that we conduct here in our institutions, at least it's very easy for us to do antimicrobial, because we just need to go and look for these pure isolates. I have to buy the pure isolates of these organisms and use them. So it's easier that we can do that. It's easier that we can do anti-diabetic study. We can easily purchase alpha amylase enzyme. Yeah, you can also buy some of these enzymes. They are available too. You can easily purchase. I think I've done a work using alpha amylase and alpha glycosidase for diabetes. You can also do some cheaper work we can do around here in vitro. You can do anti-inflammatory work on in vitro. We have done such work for somebody. We just have to purchase the COS2 or COS1 enzyme, depending on the one you're using to work. So these are some of the common in vitro research that we can do. Okay, of course, we are familiar with the antioxidants, very common. The antioxidant is very common. But the issue is that, if you do any antioxidant reports and you don't have DPPH, to me, I don't consider it to. I prefer among the parameters, let that be DPPH. But DPPH is actually expensive. So some persons just do this, the travagin effect, the peroxide, the ferric chloride, the travagin effect, all those ones. So you can also do antioxidant in vitro studies. But major major diseases, you can hardly do them here due to facilities. And these enzymes are expensive. They are very expensive for which purchase. There's a work that we designed. Okay, let me not say it because for security purpose.
Sorry, my question is this. So assuming I have a potential candidate and I do maybe MIC. So how do I know where my drug bind? How do I investigate it? Which method will I use?
The in vitro does not explain molecular level now. In vitro doesn't tell you molecular level. It is now the docking. It is now the in silico study that can now explain to you what is happening in your in vitro. Like your in vitro from your area of microbiology, your in vitro just say, okay, from your MIC, there is a minimum, there's minimum inhibition. So, but it cannot tell you the molecular interaction, it cannot tell you where it binds, it cannot tell you how the binding occurs. But when you now take it to docking, the docking now explains these molecular interactions from your 2D. You can now know the amino acids that took part in that interaction. And that was what we explained earlier before we moved to pharmacokinetics today. So in vitro cannot explain the molecular interaction. It only tells you, in vitro is a chemical reaction. You bring these two things together, there will be a change in the reactions. From that change in reaction, you can tell that something has happened. So that is just what in vitro can do for you.
Well, if I may suggest something, I think that question was asked by Dr. Adewale, right?
Yes.
I think maybe you didn't get his question very well. What he says that he has a drug candidate that has a biological effect, but he wants to find out the target, what that drug is acting upon to produce biological effect. Did I interpret that question very well, Dr. Adewale? Or Professor Adewale?
Yes.
Uh-huh. If I may, suggest, you've gotten the biological activity and that candidate drug is doing. One thing to do is that, do you know the structure of the drug, or the chemical or the compound? Do you have the structure?
Yes.
Yeah, if you have the
At
structure,
least
you can go the route of ligand-based drug design. Look at the structure.
now
Look at what.
my question now is this: does
Yeah,
it mean that all those protein that we recover from protein data bank,
yeah.
they are all simulated? They are computer aided protein.
What do you mean by that?
Does it mean that they discovered them through maybe AlphaFold or in silico analysis too?
Well, I think Dr. Henry will be in a better place to provide answer to that. He is the chemist.
Okay. See how those proteins we are gotten. We did a training on this last year October on molecular biology. Yes, I handled a training on that molecular biology. So what me has thrown it over to me. Meanwhile, that is coming from Andrew of molecular biology. Well, but then see how it all happens. Now, I have a wound. And I want to provide a cure to that wound. And I noticed that people in my area, that wound has this dominant trait, and it has that prevalent trait. So that wound, most of the drugs they have taken, the wound, the pathogen is there, is offering resistance to them, and is a concern. So what do I need to do? What we did now is we're going to take samples of that wound, see how those proteins now are got to know. They are not computer generated proteins. They are not simulated proteins. So I take samples of that wound. I extract the DNA. So if I take samples of that wound, I don't know maybe I don't have idea of those pathogens there. So I have to extract the DNA. If I extract the DNA, I will do my PCR. I will do my amplifications and all those stuffs. Then you can do Sanger. You have the amino acid sequence. When you have the amino acid sequence of that genes, you also do whole genomes to identify the variant genes, those genes that you are going to target from the organism. Then you also identify the organisms that are involved in that wound. Then once you have the amino acid now, remember these amino acids are amino acids you got from the organisms in that wound, and you have from your whole genomics and your whole genomic studies. You will not take that amino acid, the amino acid sequence that you have gotten. You now use it to model the protein, because we want to now discover a new drug for that wound. So, and we want to test those compounds first in silico. So we have to turn the amino acid sequence. This amino acid sequence we have from these organisms in the wound. We turn them to protein. We now model the protein using AlphaFold homology modeling. So when we now model the protein, we can now screen a lot of bioactive compounds to see their affinity with that modeled protein. So that is how they got these proteins. So if you also want to do a reverse job, if you want to do a reverse job now, that means I put a protein online, and that protein is for E. coli. Remember, you now want to do a reverse job, means I want to do in vitro work that we replicate exactly what I did in the in silico. So that protein you use in the in silico, look for an equalized strain, that has this similar amino acid sequence with the one you use in silico. So if you can look for that one, there are many strains of E. coli. You can get the ones that have similar amino acids. You can know from your NCBI, because even protein that band PDB provides information of the sequence. So if you look at the sequence in PDB, then look for, I'll go to NCBI, maybe you open E. coli, look for an equalized strain that will have the same sequence you got in your PDB, because you really want to target the same gene I used in my in silico. I also want to replicate it in the lab. So by the time you have achieved that strain with similar sequence, then look for where you buy it. Look for where you can buy the pure isolate of that strain. Then use
I have a question. Before this advent of molecular docking and this molecular biology of it, how are they doing it?
Yeah.
Because I know pharmacology has been existing before those things, and we always have a mechanism of any drug. So how are they doing it?
Yes. Before this.
Sorry, how are they doing what? How are they doing what?
How are they investigating this mechanism?
Which mechanism exactly?
Yeah, this receptor.
Drug binding.
How they identify the receptor, the binding site, all those things? How are they investigating those things?
Well, maybe we are coming back to my area now. The first guy that talked about receptor, Dr. Imije mentioned his name during his presentation, Paul Ehrlich, and that was in the 17th century. And it was the one that said that, look, a drug cannot have any effect unless it binds. So we started talking about receptor not today, and we got to know about the fact that drugs bind with receptors right from the 17th century. It's a germane question that you've asked, that yes, now everybody is going to docking. Why? What was the traditional way of discovering receptor? What was done before, was to look for what we call models, right? For example, we look at we want to study a drug for its vasoconstrictive. So let's say for intestinal contraction, a drug is acting on we know a drug, for example, hastens, intestinal motility. We know from our physiology that the receptor that is responsible for enhancement of intestinal motility is a muscarinic receptor. So anytime we want to develop a drug that has muscarinic receptor stimulation, we use that model, that intestinal model. We use, I'm sure most of us when we were in university doing biology-related courses, we have this organ bath that we used to use. So there are many models that we know from physiology that these are the receptors that they bind with. That was what we started with. After that, we graduated to, isolating those receptors structurally. We have, methods of imaging the receptors and binding our ligand with the receptors using highly sensitive imaging techniques. Just the way we are doing X-ray crystallography now for compounds. So when we got to a stage, we started doing that. Then at the time, we also started tying our ligands with with radioactive substances, and use those ligands to bind with receptors, and when they bind with receptors, they glow. Right? That was another that was another era, radioligand binding studies. Then later we also developed what we call patch clamping techniques, and that was we used receptors, isolate the receptors, put them on another platform. You isolate the receptor from one platform, put another platform, and you can attach that receptor to certain electronic gadgets that will tell you, that can measure when that receptor is activated by virtue of activating ion channels. And when ions begin to flow through those ion channels, it activates those electronic gadgets, and we can see how that is happening. So we've been working with the receptor using different modalities, right? But again, the advantage of in silico is, number one, it is less laborious, cheaper, and high throughput screening. Right, so those are the stages, of development of receptor study, And again, coming back to your question on the protein data bank, there are many protein databases. PDP is just one of them. Whether all of them are simulated, Dr. Eri has, provided a beautiful answer to that. No, not all of them are simulated. And again, databases, apart from protein databases, I mentioned this earlier, there are databases that are well curated. Let's say, for example, today you're a microbiologist. You just, come around and found a new protein that nobody has found before. You characterize that protein, look at the sequences of that protein, that is the amino sequence of that protein, and you say, look, this is a new protein. And you get to PDP and say, look, I found a new protein. We'll discuss on our platform about somebody that has just done that, recently in OAU. He just found a new protein, characterized the new protein, and submitted to PDP, a protein in Plasmodium falciparum, which is, a very good one for the scientific, for the scientific world, because again, this probably is going to be target for new drugs for the treatment of malaria, which is an epidemic, especially in sub-Saharan Africa. So even you yourself can come up and develop a new protein, but that is well characterized. The good thing about PDP is that it is a well-curated database. What does that mean? There are curators that will screen, all your claims about that protein before they allow it to be deposited into the database. A database is just a bank where we collect information. So if, for example, I want to study, malaria tomorrow, I can go to PDP and PDP the data bank and extract that information from there and use it for my study. So the proteins that are in PDP are well curated, well characterized, and they are backed with scientific evidence. Having said that, there are other protein databases that are not as curated, not as strict as that of PDP. Protein databases like Uniprot, Dr. Henry has mentioned NCBI, Negatome, many of them, EMBL, the protein data bank of Japan, DDBJ. So there are many of those ones are not as strict as PDP. For these uncurated protein databases, one other feature is the fact that, yes, today you can identify protein when you are working, in your wet lab. But today, because of the complexity of the genome, people are also coming up using sequences that they have found out on the genome to predict to formulate proteins based on these genomic sequences. These proteins have not been characterized, they have not been backed with evidence, but these are just predictions based on the information derived from the genome. And that, that the PDP does not correlate such proteins. If you want to find out such proteins, you go to other databases that don't have that strict, how do I call it, requirement for protein to be deposited in them. I don't know if I've answered your question.
Yes. I really enjoy your explanation, in fact, because we need to be discovering some things, all these basic principles. So we need to know them. Those are the things that guide. One of the problems that we have in Nigeria is we did a lot of theory, and that's why we are not discovering things, because we just be cramming things. People like me, after my PhD, I've made up, I determined that I don't do theory again. I'm here for for real things, practicals. If you are explaining anything to me now, you have to do practical. So I understand that one more than carrying big textbooks, unless it is necessary. So thank you, sir.
Talking about the challenge of finding a target for your drug, as I was trying to explain, if you know the structure of your drug, you can go along the line of ligand big drug design. If you look at the structure, look at other drugs that are in use that have similar structure with your drug, and those drugs, look at the targets, and of course, where to start from, which is the advantage of in silico, is to try and see if you can, if those drugs, if your own drug too can dock with the targets of the drug that are similar to your drugs. You won't, at this stage, go to the lab and start looking for and start doing wet labs in terms of using your drug, to target those receptors using your wet lab. So by the time you start doing that, you can start narrowing down on your target. And by the time you have a good docking property, then you can go ahead to the wet lab to validate it. I don't know if you can look at that route. Maybe it should be useful for you to solve that problem.
Okay.
Well, I really want to appreciate Dr. Daniel Adewale for such a insightful question. And I'm interested in your last comment that you are more interested in practical. Yes, so but make sure you have money in your pocket too, because practical is money.
Well, we still rely on Oyibos.
I
We
mean,
rely on their grant. At least we can
yeah,
explain our proposal in a layman term, in such a way that they see that our proposal is feasible. So maybe one day
yeah.
we get grants and we make discovery. So
So, you very
you're
soon,
dropped now.
yeah, very
Yes.
soon, you will stop giving us money. Because we have been collecting a lot of money. Somebody said for you to take small millions from one white man, bring to, you will sweat, you stay all night wake, you just stay awake, just make sure that you convince you want to give you money.
I observe, if you observe what we do in Nigeria, I'm a molecular
Uh,
biologist. So, and I
mm,
observe we do a lot of prevalence studies, and then sometimes we just have isolate, we send it out. You did not get a grant. Even if you have the equipment in your lab, let's say you are having whole genome sequencing, all these NGS of it. You know that it's more cost effective. If you send your sample out for sequencing, it's even more cost effective than doing it in your lab. Because to maintain the lab now, you have to have a stable electricity, even maintain
yes,
-8. If you
yes,
have a
yes,
freezer, -80 freezer, you need a constant supplies of electricity, or you have this alternative source of electricity. So that's why sometimes you prefer sending it out. And this thing, if you are a lecturer, it doesn't help, because you need to pass down the knowledge. You need to expose them to practicals. So when you begin to be sending things out, so when are you going to impart knowledge? When are you going to train people? And if you don't train people, you end up graduating Yahoo boys. They will finish from school. They start doing all this Yahoo. Thank God this we now have a computer aided
Uh-huh. So that is true.
simulation.
It,
So,
it's
but
just
I think
it's just a similar case I noticed last week. I traveled to one of my neighboring schools around, and you can imagine a student in chemistry final year would not differentiate between a test tube and a boiling tube. Chemistry final year. So I looked at all this. I said, I don't know what. Well, thank God we are now having lecturers who are result oriented, people who wants to train students in the lab, because science is not just you just come read book from year one to final year. Subject them to reading book and give them first class as well. Right.
uh,
So we want to, yes, do you have questions? Okay, Victor.
Yeah, sorry. I I want to ask, I've forgotten the name you call it, the first checking for the binding site, when you use Discovery Studio to do it, to combine two of those active compounds that has the binding affinity. That last week you mentioned that if you have like 3 or 5 that has get a greater binding affinity, you can combine all of them together in the same binding site to know whether all of them can fit into the binding site. So there was a question I asked one certain time about interaction, you know. So there was a work I did, but it's just more of a plant, a bio, a compound in a plant having interaction with a known compound like a drug. So does it mean that you can use this avenue to check the interact? Because I'm just interested in interaction study, because most of the problem you're having with taking herbal medicine or even plants is sometimes you take them and you may take a normal drug, but you may not know how the interaction will be, whether the interaction happens. So I just want to link it with each liquid so that I can visualize it, and before I create ideology in the lab, in the wet lab. So thank you very much.
Okay, I think let me relate it. What we did that day was a superimposition. Yes, where we superimposed our own ligand with our test ligand with a native ligand to see if the interactions actually took place at the same binding post. So I think that was what we did, and I have that same image. So, but I have a second understanding of your question. If I get it clear, this was what we did, where we see if the ligand on red is interacting very well with the same active site with native ligand on yellow. So we saw that they were well super imposed. That is for docking validation. But I also have another understanding of that question, which is a drug interaction. Yeah. If I take 2 different drugs now, don't I think that these 2 drugs might interact by themselves? So I don't know if that's also part of what you're asking.
Yes, sir. My own is herb and drug interaction anyway, but it's the same drug
Yes,
and
yeah,
herb drug.
drug interaction. Even the herbal I'm taking too, the various components, don't you think they might interact with themselves, and then that interaction might lead to something else? For now, one of the only way you can do drug-drug interaction, we have part of those parameters. We have part of those parameters under metabolism. No, under metabolism, or the ones on metabolism. They were telling us if there could be drug-drug interaction, even some on distribution. Yeah, some on distribution too. Also had idea if there could be drug-drug interaction, but they are just assumption parameters. Now, you can know if a drug can interact with another drug when you do ligand docking. You know, we are well conversant with protein ligand docking, but we have a ligand docking, we have protein docking, and we also have a protein ligand docking. So if you want to study how a drug can interact with another drug, then you go for ligand docking. Get some of these softwares that can do that are expensive. I always recommend MOE. If you can get MOE. It can help you to perform ligand docking. Otherwise, you just rely on the distribution parameters that tells you if there is DDI risk. You know, I mentioned DDI risk, which is a drug-drug interaction, as part of our distribution parameters. So you can rely on those probability values that tells you if your drug has the ability to interact with other drug. But if you really want to visualize the interaction of a drug with another drug, then you go for a higher sophisticated softwares like MOE. They can do such kind of interaction. Yeah, thank you very much. I'm actually interested in drug-drug interaction because it's part of the factors we need to consider. You know,
Yeah,
we will now
sir,
start.
you said something sometimes. So, and I don't know when we are going to implement it. When
To do
we
what?
discuss about protein docking using Schrodinger or MOE, you said that some of them are paid, and you said maybe sometimes we can contribute
Yes,
and having access to
yes.
it. So I don't know. I want
Unless,
to ask.
yes, unless what I can do is, I think so far we have trained more than 1,000 participants. Yeah, so far we have trained more than 1,000 participants so far. And a lot of them are interested in sophisticated softwares. So unless we can come together, pool our money together, see how we can get one or two of these expensive softwares that can do better things for us. If we can achieve that, then we share the license to as much as people who contributed to that. So that is the only way out we can do to get some of these softwares. So imagine as of 2021, MOE was at $3,000, $3K. So if you want to calculate $3,000 now, you know how much in naira that we are going to spend. So you know the equivalent in naira now for $3,000. So how much can we share within ourselves? So can one person now sacrifice 500,000 naira to get the software? tell somebody to bring 500,000 out to buy software. The person will the person will tell you, oh my God. You know what person will tell you? To pay 500,000 naira to buy a software. Maybe we are 10 or 20 or 50 and we are paying 500,000 naira. Maybe if we are 10 or if we are 10 of us can generate up to 5 million. Yeah, and it
I want to ask.
can go.
I know some of us, particularly, I know people like you. You are already experts in this area. Maybe you are using one of them. At least if you can just can we just support so and you share your own so with us.
Yes, like I'm a very sincere facilitator. I don't hide knowledge. I don't hold knowledge. I made it very clear that the license for MOE expired and we could not afford it again. So even myself, I don't use MOE again. Some persons have screen digger. You can manage screen digger. Screen digger is another sophisticated software. So I think the training we'll be doing a March. Yes, the March training. We'll be using screen digger for that training on March. That is the training on the Pharmaco 4 and bioisostatic test screening, molecular dotting, and other one. So we'll be using screen digger for that dotting aspects of that training. So you can join that training, and of course that training will be a wonderful one. It's power part. We will learn, we model by ourselves, we stream by ourselves, we build our structure by ourselves. We make sure we call it de novo modeling. We create a compound, do the screening.
Hello? Can anybody hear me? See my network. Hello? Hello?
I think he's having poor network.
Okay.
Good evening, Prof. Utimi. I want to ask, this is the person, I don't know whether your department, AU, whether they do bioinformatics or chemoinformatics in your school, in postgraduate studies. So I just want to know, because I think there is no much school that do it in Nigeria. I just want to confirm, sir. Thank you.
Sorry, we don't do bioinformatics at all, at any level in OAU. We don't. And of course, that has been a challenge, not just in OAU, the whole of Nigeria. In Nigeria now, I think the only university that I know that is doing bioinformatics with as a degree or as a course in Nigeria is only Covenant University. Covenant University is in Ogun State. The church that, the university that is owned by the by winners. So that's the only university in Nigeria that does bioinformatics at the master's level, and I think they have it to the PhD level as well. But the only caveat there is that you have to be full time and you have to be fully on ground for you to do it, which is actually a challenge. I've also been looking for, universities within Nigeria where one can have a degree in bioinformatics, and unfortunately, that is what we have for now.
Hello, sir.
So yes, somebody is trying to provide more information on that.
I think we just started a while ago. They are doing what's it called, a postgraduate degree in bioinformatics at master's level and PhD level.
Is that
Uniben does it at master's and PhD level.
One thing is to be doing it. Are they really doing practicals? Let's ask those questions. Are they really doing practicals?
Yeah, they are. Like Dr. Vincent said in one of the sections, they are trying to, put up a bioinformatics lab, right? In Uniben, the faculty.
Are they maintaining it?
Yeah.
Well, if you look at Covenant University, in computer science generally, they are doing fine. I think they are even number one in the whole. One thing is you buy computers. Do you even have personnel? Do you have electricity? So do they even buy the quality computers? So they just use it to decorate for the purpose of accreditation.
Well, the bioinformatics program in Uniben, I've asked Dr. Emeji about it. It's not a full bioinformatics program. It's a farm chem program, and they are trying to introduce bioinformatics into it. So it's still an MSc farm chem with options in bioinformatics. So it's not a full bioinformatics course. If you are interested in that, you can try Uniben out. But if you are looking at a course that is purely bioinformatics, the only place that you can do it for now is Covenant University. I've been on this for the past 2 or 3 years, and that is the fact. I've talked to Dr. Emeji about the one that they are doing in Uniben. It's a purely farm chem degree, but with options in bioinformatics. If that suits you, you can link up with Dr. Emeji.
Okay, sir.
Yeah, sorry, my network kicked me out, so I had to come back. I really want to appreciate us so far for our contributions, our questions. So we have come to the end of Pharmacognosy, and it has been successful. So, Victor.
Sorry, I'm asking too much question today.
No. That is it.
Okay. I was going through a paper today. So I saw how the site is shared PubChem, very broad. So I don't know, all those things. Now, how will you cite, for example, now if you are using different in PubChem, if you are using different CID, that is different molecule. Now I need to cite them, like each of them now. So assuming you are using because they have different citation. If you're using different things, so how will you cite it? Will you cite it like PubChem generally, or will you cite different edition? Because some of them cited those different articles individually. So just what I
Okay,
wanted to
no,
know because you're going to write.
no. If you use pop cream, maybe you use pop cream to do whatever you did too many things in pop cream. You downloaded, you retrieve different structures in pop cream. In as much as you use pop cream, you cite pop cream, and where it will appear in your in-test citation is in your methodology. So it will be in your methodology where you stated the method you did. Probably structural data files were retrieved from an open source server, PubChem. You state the date, July 2026, where you assessed the date assessed, you state it, and then you put your citation depending on the citation format you are using, the reference format you are using. So you cite just the PubChem generally. Then you come to your reference page and also put it there. So you don't need to cite separately based on each of the things you performed. Yes. And most of us, we don't take this citation serious. One day they will do it to us. Any web server, any web server you use, even if you use Pirates, Pirates has think you cite Pirates. Yes, Pirates has citation. If you are docking with Pirates, you will see once the docking is going, you will see the citation. That should be throat and awesome. Throat and awesome. You will see the citation there. You copy it out. Why that docking? Why the thing is showing running in Pirates? Why is doing running as it's docking? The degree is going. You will see the citation up there. You copy it and cite. Since you use Pirates in your docking, you cite them. Then put the full details in your reference page. So whichever software you're using, you cite. I don't think if Discovery Studio we have, but you can now cite. You can cite. I think if you carefully look at Discovery Studio, we may see citation, but that one is not a method now. It's just a tool you use. It's a software you use. But I know that Pirates has citation for docking methodology. So any web server you use, their methodology you cite them is very, very important, and that will make them know that people are actually assessing their site. The same way you like people to assess your work and cite you. So thank you very much for that, Kristen. So
Hello, sir.
the platform is open to everybody.
Hello, sir.
Sorry,
Yeah.
we are still for the training that was held yesterday. So your promise has not been fulfilled. And
Yes.
the
Yes. Yes. Everything has been done. We shall be. I promise that it will be today. My promise was before this in the evening, but promise failed. Don't forgive us. But I'm going to send that. I've received a slide from our keynote speaker, and already we have prepared our video already. So I will just have to compile the folder and drop the link this night. Failing me, please. So the yeah, Dr. Daniel,
Yes, sir. Thank you,
yeah,
sir.
Dr. Vincent was telling me about you last night.
I didn't get you. Say what?
Dr. Vincent was actually he mentioned your name in our discussion last night. Go
Oh,
ahead. Mm.
okay. I want to ask concerning that with the interaction that you mentioned earlier. When I was doing some of my own ligand protein complex, I noticed what was it called now on favorable acceptor interaction. So I was curious as to what that meant, and it wasn't part of what you said. So I was just like, what was this? Does that mean, the ligand interacted properly with the protein, or I don't know.
Yes. You will see them on red, right? On red.
Yes, I read.
Yes. It's normal, depending on the interaction. They are just unfavorable bonds. But in as much as you also have a conventional hydrogen bond interaction, most times carbon hydrogen interactions and van der Waals others, you can ignore that unfavorable bond. Where we pay attention to that is when you did when you visualize the interaction and you notice that there was no conventional hydrogen bond interaction, and then there was an unfavorable bond, then we have to reject that interaction because there was no conventional. Every interaction that does not have conventional hydrogen bond interaction, just throw it away.
Okay, so
Just throw it away. That's my number one concern. If I look, I do not see that conventional hydrogen bond interaction. There's no need. No need for me to go further. I just have to modify. Sorry, I said 20 away. Don't throw them away. Oh. They are useful. Just modify them and proceed with your work.
oh,
There is, you don't have to be scared with that red unfavorable interaction. I've seen it countless number of times.
okay, sir.
Even Steph, there are some things that as you begin to proceed, you encounter this challenge on your own. Like somebody or was it you that sent me that message? No, not you. Not you. Somebody sent me a message on WhatsApp. Or is you?
About what, sir?
That should be about something showing ligand, not a single fragment.
Yes, sir. It was me, sir. It's me.
Okay. So such challenge now. Have you been able to overcome it?
Yes, I deleted that first end, that first chain. Then it worked.
So such kind of challenge now, you may encounter them and you have to do that, figure out a way on your own. Even too, there are sometimes you may want to convert your protein to a macromolecule to an orthodox macromolecule. It will tell you conformations found, coordinate conformation A, B, conformation C. So when you encounter those things now, and then you have to find a way to figure out because definitely they cannot start preaching everything from A to Z. But I bet you there are a lot of issues you're going to encounter once you proceed in doctrine, which you have to find your way out. Most of the things I teach, we are not what I was taught. In fact, the things I teach, if I want to teach based on what I was taught, I'll end up teaching like 20%. So most times what constitutes what I teach are now my personal experience, my personal research, and I just felt, can we try this thing? Like what we do in the office is we keep asking ourselves questions. Can we try this thing? Can we turn this thing to this way? And if it works, I have to now teach the public. It will now constitute part of my training. We are not taught that way when we are taught, but now we are not seeing new way. So we have to declare it out and teach people. So the same way too, me that claim that I have encountered some challenge and I have solutions, there are some challenge too that you keep experiencing, and You just have to look for a way to find solutions. There was a solution I had, a problem I had to go to YouTube, no way. When I started nanotech modeling, yes, when I started building nano complexes with my laptop, up till now online on YouTube, you will not get any YouTube video, because even that video, I've not uploaded those videos. You won't get YouTube videos that is talking about how to use your chem draw, your chem sketch to build nano complexes. You won't see them, but I did it myself.
Yeah, I think you said something concerning the beginners during the beginners course that you can use ChemDraw to do nano
Yes,
stuff.
so I did it myself. I will have a training on nanotech coming soon. I don't know the month, but it's part of our calendar. I just sat down and figured a way out. I have done it for a lot of people. People have published papers on that. So you acquire more knowledge on your own.
Okay, sir.
Yeah. Once again, I think at this point we are well relaxed and successfully have ended Pharmacognosy. So do your work. We are agreed of is it a month time? Yes, we are agreed of a month. So you have from that to 1 month for develop your manuscript. Develop your manuscript, very important. Without that, we said no certification. So just try your best, and I believe most of us are even done with their work, because some persons we are doing it as we are doing. They are doing it together. Why some persons wanted to wait until we finish, then they will not start their own. No problem. I don't know, like you forget too many things. But thank God for the video. So we have lifetime access to that our video, the drive. So you can come back to it at any time. So I want to appreciate. I will even encourage you to go and download them. Just small data. Download them, keep them to your phone. Or even if you not download them, they are saved there. Anytime you come back, you go and watch them. So I want to thank everyone. We had 70 registered participants for this training, and that was massive. And but here we are, 15 left. So there are some persons who registered but never attended one day. But thank God that they are following up with the video. At least some of them will come to my DM and ask some questions. I have to respond to them. It's part of the training. So I want to say thank you for all those who shared our adverts, especially Uniben. I've told Dr. Vincent that I owe Uniben a gift. I said it when we started. Uniben, we are well represented. So I think I'm planning of coming to Uniben very soon. We have a plan of coming to Uniben. So for those of us who have joined Green Chemistry, it promises to be wonderful. Yes, it promises to be awesome. All our trainings are carefully planned. So by next week, we have a free training on QSAR and QSPR. Then by last week of this February, we have a beginner's course on data analysis. You know, as scientists cannot do without data analysis, so we'll take you to the foundation of data analysis, some of the basic things you need to know, and get some data, see how we can manipulate those data. While we are preparing for Eve Macro 4 modeling, by March. So the flyer is already out, but we shall place the advert tonight. So encourage your people to register. Those in pharmacognosy, medicinal chemistry, pharmacy, biochemistry, molecular biology, bioinformatics, technology, chemistry, they are all welcome to this training as we teach them how to do de novo modeling, build pharmacophoric features, and then see how we can target them. And one of the things I want to ship into this training by March is retrosynthesis. How can I be able to show them how to synthesize this compound we have modeled? I think it was not part of the module here, but it just came to my mind as a giveaway prize, as a giveaway token. So we introduce it to some softwares you can use for synthesis without you cracking your brain, trying to plan the synthetic pathways. So while we wait for contact on where to do synthesis, we can just design the synthetic route, the mechanism by ourselves. And there are still some synthesis we can still perform, some common synthesis that we can still perform, just like those ones with a Friedel-Crafts acylation and Friedel-Crafts acylation, the ones that involve electrophilic substitutions and some nucleophilic substitution reactions. We can easily perform those ones in the lab with high precision. So because I've also done a lot on that area, and the product was really what we wanted to synthesize. So I want to appreciate every one of you for pulling through to this very moment. As usual, I will take my time to appreciate everyone at this very moment as we draw the curtain close. Some of us are students, undergraduate students here. I really want to appreciate them. Mostly, I want to appreciate Abisoye Dolphin.
Thank you, sir.
Yes, I really want to say thank you for always being around. I appreciate Abiodun for being around. Oluwatoyin and my friend Dr. Chioma Wofo. I also want to appreciate Dan Ademole from Bowen University, and Dan Irene. I was at Bowen University though for an MBGN conference. Okay. I want to appreciate my friend from David Umahi, Dr. Daniel Oruke, and Dr. Excellent. Currently, I don't know what happened to David Umahi. I'm seeing a lot of participants from that school. I don't know if they had meeting. Did you guys had meeting?
On what, sir?
Oh, almost. Because I wanted to ask Dr. Excellence and Dr. Daniel if they had meeting at that school. Because
about what now?
I'm seeing a lot of participants from that area.
Okay.
I did a work for somebody, Dr. Aja. She is from your school.
Florence.
I don't know,
which of the Aja?
Is it Abi or Obi?
Obi. Okay, Aja Obi. Florence.
Yeah, something.
She's
Obi.
my friend.
Oh, I just finished the work for her.
In biology.
Okay, because that work was on homology modeling. I modeled some proteins and did the docking for her. So I want to appreciate Professor Justina Otombo, Udotong. Yeah, Udotong. And I have a friend from Bo University also. She has always been active. Olewoye Bolanli Mary. Sorry if I destroy your name. I am an Igbo.
Thank you, sir.
Yes, I'm an Igbo. I'm from the east. Sorry, I'm from the south, precisely from River State. But I spent all my life in Igbo land. I
You're welcome.
appreciate, yes, I appreciate what me, all I be. Thank you so much, everybody. Obo Chioma Messi and Victor Amoke. So I really want to sincerely appreciate all our effort. You know, it is not easy coming online, being available every class. And mostly Dr. Olabi, who is not, I think wherever he is, he's very busy over there. But he comes up even before the time. So I wish I can extend my Valentine gift to everybody.
Please do. Please do. We can't wait to receive it.
So in what form do you want the gift? Valentine gift, in what form?
Training form now. Give us
Okay,
one package of paid training for free.
so I should have a training
Maybe the
on
match
that
code we
Valentine
can
Day,
just
right?
maybe one month intensive one.
Okay, 1 month free intensive training.
Yes.
As Valentine gift, Jesus. It's well, I'll be giving discount to those who wish to join the March training from this macrognosy training. So if you wish to join the March training, just DM me. I'll give the discount. And currently now we are appreciating those who are referring their friends. So we are saying your referral should not just be in vain. So we are giving 5% discount because we notice that people have been referring for free. Like some persons recommended a lot of persons to join this training and they paid for it. So why won't we appreciate you? Because we never asked you to do that and you did that from your will. So we want to show concern by giving out 5%, 5% to 5% of whatever you pay will give back to you for every one person you refer. So to every one person that you refer that join the training, we'll be giving you 5%. That is starting from the March training. So if you're able to get us 2 persons that paid for the training, then we are giving you a 10% referral bonus. So we are doing that to just encourage those who constantly refer their colleagues to our training. I think that is a bit okay to encourage us the more. So imagine you now refer the 20 persons, 5 times 20, 100% discount. So the money you are paying will be given back to you. So it's just a way of telling you thank you. So once again, we say thank you. So continue your research work. We have 1 month after today. So probably today's date is 12th. So by the 12th of next month, we are expecting to have our project defense. Our project defense by around 12th of March. So that 12th of March should be on Thursday. Yeah, because Tuesday is 10th, Wednesday 11th. So yes, that 12th also will be on Thursday. So say good night to everybody. Thank you very much.
Okay. So, sir, please, have you been able to decide on what we asked you for? The slides of that last year's November, sorry, the
Yes,
videos.
they haven't given me
Say,
approval. They have not given
please
me
let them
approval.
do now.
I have talked to them and
still talk to them again, please.
Yes, they haven't,
Even
but
if it's for some few days, okay, so that people that want to download it can download it.
okay, like how much do you think people can pay for it?
Maybe it might be the Valentine gift, sir.
Okay, I think so. We'll do that as Valentine gift. So you buy enough data to
Okay.
download all the video.
Ah,
download the video.
it's okay.
Once
On
you
14th. I just
once you,
just tell us on time. So at least we'll
yes,
start downloading it.
I will. So I think we'll do that as Valentine
Okay.
gift.
It's
Yes.
okay. I appreciate.
Once again,
We
we
appreciate.
appreciate everyone. Thank you
Thank
so
you.
much.
It's okay. Thank you, sir.
Good night.
Good night, sir.
Good night.